Okay, I think it's almost time to start the seminar. Okay. Good afternoon, everyone. My name is Gi-seok-yoon, Keralics Material Transformation Divisions. Today, I'm very happy to have a seminar with the Professor Hideki Hashimoto. He's from the Osaka City University. As you know, where is located Osaka City University? Yeah, it's located in Osaka, but in the university, I know in there is do you know the 2 Nobel Prize winner scientists produced in that university? Actually, the Shinya Yamanaka is get a PhD from that university, and what's that? His name, the Nambu, the physics. He's get also the worst professor in there, and Professor Hideki Hashimoto. He was the the professor and project leader of the Osaka City University Advanced Research Institute for the Natural Science and Technology. He got the PhD from the Kansei Gakuin University, 1990, and then he has the many experience the academic career and then particularly he was experienced the visiting associate professor in the University of Tokyo and the University of the Glasgow, Scotland, and United Kingdom. And in 2002, he became a professor. And then among his research career, he highly studied about the focus on the photosynthesis, particularly the rice harvesting. And recently, he studied on the artificial photosynthesis. Today, his title of the lecture is the Artificial Photosynthesis Production Solar Fuel Cell. And okay, before I talk, and also, do you know he's actually the professional martial art, the expert, the karate. So anyway, please start your lecture.
Thank you very much, Professor Yoon, for very kind and nice introduction for me. And it is my great pleasure to come here, and thank you for inviting me to come. And today, I want to talk something about the artificial photosynthesis, the state of the art of those things. And I'm trying to explain, not in very details, but I would like to have rather broad and broad perspective, what is going on in this research, very hot research field. Okay, this view graph shows the world total primary energy supplies. And as you know, that the 80% of the energy is supplied with fossil fuels like oil, coal, and natural gas. And more importantly, the 80% of those are used as they are. Okay, so people usually think about the energy is almost equal to electricity, but actually that is not true. We need to have a real fossil fuels. And the big issue right now to come to us is that we need to have new renewable energy sources, but how soon are they needed? At the projected consumption levels, the supplies last approximately, it is called in 5 years ago that the coal is 230 years and oil is always 45 years and gas is 60 years. And even nuclear fuels, if we still can use that nuclear fusion reactors, the natural abundance of the radioactive uranium is very restricted, so it's only percent for 55 years. But that is the estimate 5 years ago. something has changed. For example, because of the highly industrialization in China, they use huge amount of coals every year. So now the estimate becomes less than 200 years. And, but fortunately, in the United States, they have found new natural gas sources called the shale gas. So because of this revolution of shale gas, this 60 years becomes 160 years for United States, okay, not for us in Japan. But of course, those are the sort of the pessimistic point of views, and those figures of numbers cannot be the double that to get to the optimistic view. And even more worse thing is that the world energy consumption is predicted to rise with growing population and industrialization in developed countries by 2 to 3 times in 50 years. Then that accelerates to burn the fossil fuels. Then eventually the rise of the CO2 levels with its severe consequence will stop us, need to stop us using fossil fuels long before they are used up because of the global warming. Okay. Then how shall we do? That is the issue. And if we look up, then you can find something very shining things. That is solar energies. As you may know that the amount of the solar energy which hit the surface of Earth for 1 hour, only 1 hour, equals to the amount all the human beings using within a year. So sunlight energy is really vast and enormous. And actually, now we have good technologies for solar batteries. And even for the commercial solar batteries, the efficiency of solar energy to electricity conversion is nearly 20%. And if we, for example, to spread those solar batteries to the most bright part in the Earth, then these red square parts, if they are filled with this 20% commercially available solar batteries, we can produce 20 terawatts of electricity. That is good enough. But the issue is how we can store those electricity, such a huge amount, and or how we can transfer those electricity to all over the world. In Japan, we have a very nice, electricity transferring systems, but that is not actually very true. For example, in United States, maybe 40 or 50% of the electricity has been lost as a heat during the transport. Okay. Then what we need to have is a real fuse. Then how we can make it? Then if we look back to the histories, and actually we know there is a good organisms that can produce some organics using sunlight. That is a photosynthesis. The photosynthesis is divided by 2 reactions. One is light-dependent reactions, and the other one is carbon fixation reactions. The very smart point of photosynthesis is they can use the solar energy with a very efficient way, and they produce the necessary electric motive force to proceed several catalytic reactions. The important thing is they can split water and produce electricity and protons. Then, in the dark side of the reactions, using the Calvin cycles, they can fix the CO2 and produce carbohydrates. Actually, this is a part of the fuse which we really want to have. If we look at the real photosynthetic reactions more deeply, then this is a sort of the cartoons which explain what the thing is going on. There are many pigment protein complexes that has good functions connected to each other, and then produce energies that are necessary for those photosynthetic organisms to grow. Actually, for the right reactions in PS1 and PS2, two types of photosynthetic pigments. One is called carotenoids, and the other one is called chlorophylls. They are deeply involved to capture the solar energies. And then, utilizing the energies, they split water and produce electrons and protons. And in dark reactions, there are several types of the enzymes. For example, there is a hydrogen-generating hydrogenase type enzymes. If we use those, then we can produce hydrogens, or they can, of course, synthesize carbohydrates. And if we can modify some part, maybe this can be go through to the production of ethanol, or in the JB in the United States, my good friend Jay Kiesling, he's working on how to produce biodiesels using this type of bionic organisms. Okay. But this is relatively slow and less efficient processes if we think about overall efficiencies. Only the 2 to 3% may be converted to the things which we need to have. So the definition of artificial photosynthesis is a sort of the things which can accelerate the reactions to produce carbohydrates or hydrogens as we really want to have in the real time. To model those reactions, actually, this is the scheme of photosynthesis. In the case of the photosynthetic organisms, there are many types of enzymes are related to each other. But the essential part is they have two reaction centers that produces electromotive forces. And then they can connect in a very good way and produce high electric potentials. And so if we mimic this kind of the reactions, maybe the image of artificial photosynthesis can be like this way. We need to have reaction centers to produce electromotive force, but to drive that, we need to have a good antenna systems which support the enough amount of the energies to the reaction centers. And this reaction center should be conjugated to two types of catalysts. One is oxidative catalyst, and the other one is the reduction catalyst. And oxidative catalysts should split water to produce hydrogen, to produce electrons and protons. And the reduction part that can synthesize the necessary fuse. That is a very ideal systems. Okay, to make feasible to this kind of research more efficiently, in our university, we have founded a new research institute which that Professor Yoon has introduced me, but OCARINA, Osaka City University Advanced Research Institute of Natural Science and Technology. I named this. OCARINA is a very memorable and very good, This is a sort of the music instrument. And in the ancient book, it says that is the tools of Solomon's. Okay, so which can, what should I say, fascinate devils or something. Okay. And since we as a city university, we have very strong relations with Osaka City. So the main purpose of this research institute is urban studies that include renewable energies. And this institute was founded as the Distinguished Research Institute for Photosynthesis and started from 2011. And we also promote the University Business Corporation. For these purposes, I have asked to the former mayor of Osaka City, Hiramasu-san, not Hashimoto, and he gave me how much? $10 million? He gave me $10 million to build new research buildings for Research Center for Artificial Photosynthesis. And we aim to target the commercialization of the solar fuel productions until 2030. I will show you the roadmap later. And actually, Professor Young had visited me when 3 weeks ago, 2 weeks ago?
2 months ago.
No. Only less than one month ago, he visited us to research this institute to do the joint research. And this is a very interdisciplinary research institute. Physics, chemistry, and biochemistry research groups collaborate with each other. And all the students and academic staffs are living in the same big room and discuss always. And we have nice facilities for human food and also biochemistry lab. And actually, this is the most expensive part. This is the ultra-fast laser systems room. And yes, actually, our university has been founded a very, very long time ago. And actually, this type of buildings are the sort of the heritage, national heritage type buildings. So we refurbished all the inside of these old buildings, and we have a very nice facilities for the joint research. And actually, this is the Research Center for Artificial Photosynthesis. I named this as a recap, like name after JCAP, KCAP. Our institute is recap. And this is the photograph of the opening ceremonies. Probably you may know this guy. This guy is Professor Nobuo Kamiya, who has determined the precise structures of PS2. And this is me. At exactly the same time, I have a class to teach karate for university sports class, so I was watching from there. So the targets, of course, our target is the production of solar fuels, but at the same time, we also need to think carefully about the solar battery, although the storage of electricity is a big problem, and also the electricity cannot be directly becomes a carbon source. And hydrogen evolution is maybe your interest and also our interest. This is one of the most simple form of the energies, but still holds a similar concern with solar batteries like storage, and this cannot directly be the carbon source. And our final goal is the production of methanol. Okay. Since if we can synthesize the methanol using the solite, the issues for artificial photosynthesis and the shortage of fossil fuels can be solved altogether. And methanol is the most simple organic compounds. And also, this is very important. The current infrastructures for gasoline stand or something can be used. And also, the transportation itself is not very difficult because methanol is liquid. And yeah, methanol immediately can be used for, because Indy 500 automobile race has used methanol as a fuel until 2005. So if we have a good engine systems, which can run with 400 kilometers per hour. That is good enough, for driving cars. And this is also important, the Toshiba has already been invented the direct methanol fuel cell. And the aim of artificial photosynthesis can be summarized like this way. It is the substitution of rare metals such as platinum and ruthenium. And I will talk you later that including the light harvesting systems that can concentrate low density of solar photons is also very important. And one of the most difficult point to get over is the splitting of water to produce protons and electrons. If somebody do this, then the world may change. And of course, if possible, maybe hydrogen production is at first, but if possible, we want to use the CO2 as a carbon source. But right now, searching any possibilities is the most important. So all the new researchers are required to be involved, and also the students and young investigators need to come. So we need to expand this research field. And our strategies for artificial photosynthesis like this way. One is modularization, because I showed you artificial photosynthesis can be split into 4 parts: light harvesting, reaction centers, and 2 catalysts. So we need to modularize and aiming the higher function and the diversity of each module. And to select the catalysis is very difficult and important. But the most important thing is screening. We can synthesize and produce many metal oxide catalysts, but screening technique is very important. And I think that the electrochemistry can be a good candidate to solve the issues of the overpotentials, and as well as revealing the mechanisms of catalysts. And combining all these modules, we need to produce solar panel-like devices. That's what we want to do. And actually, this is a roadmap of artificial photosynthesis producing solar fuels, which I made a presentation at the City Congress of Osaka City. And all the council members believed me. So they have decided me $10 million. Okay, let's go to the science part. First, I would like to show you the recent achievements and the state of the art of the light harvesting systems, because this is my main concern. Okay. Why light harvesting system is necessary is if we calculate, for example, the density of the solar photons, solar energies at the surface of Earth is 1.37 kilowatt per square meters. This is well known as a solar constant. And if we convert this number to the photon numbers, then that corresponding to this energy is just slightly greater than 6 times 10 to the 21st per square meter per second. And this is all photons. The photons which we can use is in the visible light regions is only 1.6 times 10 to the 21st. But this is looks like a huge numbers. But if you are the chemists, this photon number corresponds to only 1/400 of Avogadro's numbers. So if this solar radiation can split water with 100% efficiencies, that only split 11 milligrams of water per square meter per second. This is good enough or not, depend how you manage the systems. But we need to have a good light harvesting antenna to concentrate all the necessary photons to drive the catalysts. Okay. Otherwise, huge area is necessary to make a good performance of artificial photosynthesis. If we compare the solar radiation spectrum to the spectrum of the various kinds of natural light harvesting systems from photosynthetic organisms, they show this type of good correspondence. So natural photosynthetic light harvesting system is a very, beautifully organized so that they can share all the energies of the solar photons. And amazingly, they can utilize almost the almost similar energies of the band edge of the silicon. Okay, 1100 nanometers. That is almost similar to the band gap of the silicon. Of course, silicon is a very, very, very nice materials, but these photosynthetic antennas also can utilize all the photons which in these spectral regions. And among them, we are now mainly focusing on the to quest the antenna systems of purple photosynthetic bacterium because it is easy to handle in the laboratories. Okay, and this shows the absorption spectrum of photosynthetic membranes called the chromatophores from one type of purple photosynthetic bacteria, Rhodobacter spheroides 241. And as you can see, they have a very beautiful, structures in the visible to near infrared regions. And all of these structures are coming from two types of pigments. One is carotenoid, which absorbs the blue-green region of light, and also chlorophylls. In the case of bacteria, it is not exact chlorophyll. Actually, that is a Bacteriochlorophyll. And when carotenoid absorbs the energies of the blue-green region of light, it transfers its energy to Bacteriochlorophyll, and then all the photosynthetic light reaction starts. It is well known that there is two types of antenna systems. LH1, which surrounds the reaction center complex, and also another type of the light harvesting systems, peripheral light harvesting systems, which is called as LH2. There is LH1 and LH2 in the purple photosynthetic bacteria. And if the light energy is captured by LH2, the energy captured is transferred to LH1 with nearly 100% efficiency. And then this energy is also goes to the reaction center to nearly 100% efficiency. Okay. The important thing to make research to reveal why this type of highly efficient energy transfer can be possible. So we need to know the exact structures of these complexes at first. Fortunately, now we know very well about the structures of those complexes. This is the structures of LH2, which has been determined by my good friend, Professor Richard Cockdale at the University of Glasgow, Scotland. And they showed us very nice, beautiful ring structures for this complex. So ring structure has some meaning because of its ring, so they can capture all the photons coming from any directions. So nature is clever enough to organize this kind of beautiful structures. The size of this complex is only 50 angstroms, only 5 nanometers. And it is, if we remove all the protein parts, the complex is organized with these photosynthetic pigments. The purple one is monomeric Bacteriochlorophyll, and green one is a dimeric Bacteriochlorophyll. The carotenoids bound to both monomeric and dimeric chlorophylls and transport captures energies in the blue-green light regions. And in the case of the LH1 reaction center conjugate, the resolutions of X-ray crystallography is not still very good enough, but there are three types of reports on the structures. One that has a dimeric cores, and one that has elliptical ring structures which has a gap that kinome molecules can go through. And quite recently, this year, which group? Professor Miki at Taitech, and also Otomo-san. Otomo-san, actually he's a Chinese guy, but Otomo-san got married with Japanese lady, so he became Japanese. He decided this type of closed ring type LH1 reaction center structure. So this is the newest one. Okay. But important thing is these ring structures are composed of the several monomeric units that is composed of two types of alpha felix and photosynthetic pigments. So very simple. Okay, with using this type of simple structures, they can modulate the energies of the absorption bands of each photosynthetic pigments, and they try to cover the energies in the whole visible and near eye regions. Okay. Important thing is, as I explained to you, the energy transfer efficiencies between LH2 and LH1 to reaction center is nearly 100%. But the efficiency of energy transfer from carotenoid to bacteriochlorophyll molecules are not 100%. In natural systems, they usually have regulation systems. Since in photosynthetic organisms, they have started to grow in the deep sea, and in that time they may have a good efficiency of the photosynthesis. But after they moved up to go to the land area, the sunlight is too strong for them. So they need to adjust to release excess amount of light energies. For that purposes, I don't know, but since I am a researcher on carotenoids and chlorophylls, and I know very much details how the energy can be transferred between them, and we did study a lot. After I have published a science paper in 2002, every year new mechanisms has been found, and actually carotenoid has the absorption in the blue-green region of light. This corresponds to the ground, to the second singlet excited states. And carotenoid has hidden, optically forbidden excited states as S1. So once the carotenoid molecule could capture energies, it transfers energy to bacteriochlorophylls from these two types of singlet excited states. And more interestingly, there are another type of several hidden excited states were found and named as SX or S star or S dagger or SICT or something. So carotenoid looks like a very simple molecules with single and double bond conjugated systems. But actually, its electronic excited state structure is very, very, very complicated. And even, for example, if these carotenoid molecules are successfully transferred to energies to bacteriochlorophylls, this energy sometimes go back to carotenoid and they dissipate, the excess amount of energies. So to achieve the really good photosynthetic reactions, for these organisms, 100% energy transfer is not always necessary. So photosynthetic antennas that nature had created have various photoprotective mechanisms against excess amount of light exposure. Okay. So it is necessary to produce artificial antenna systems that eliminated unwanted energy dissipation pathways in order to create super highly efficient photosynthetic antenna that is needed for the construction of the artificial photosynthetic systems. How we can do that? since natural light harvesting systems are composed of a relatively simple number of the elements, only the two types of the alpha helix and also the pigments, and they are not covalently bound to each other. So if we can use a good detergent molecules, we can dissolve and divide the elements to the monomeric unit, and using organic solvent extraction, we can extract only one of those pigments, and then we can get in back the different structured carotenoids in there, and in the test tube, we can form the similar type of the aggregates, the ring structures of these complexes. So we call this is a reassociation. And sometimes we can completely divide to each element. And in the case of the alpha and beta alpha helix, they are only composed of 50 amino acid residues. So 50 is, sort of the accessible amount of number of amino acids we can synthesize using organic chemistry. So we can modify the structure of this peptide itself, or sometimes, since we know all the genes of the biosynthesis of those pigments and proteins, so we can modify the bacteria itself using molecular biology techniques. And then we can change everything, the structures of those pigment protein complexes. Then what we have done here is we utilized the light harvesting systems which are produced from red photosynthetic bacteria, which called Rhodospiridium rubrum. That has a carotenoid spiroxanthine, which has 13 conjugate to CH double bonds. In this case, they only have 28% of energy transfer efficiency from carotenoid to bacteriochlorophyll. It's amazing. 70% are released as a heat, because this bacteria is living in the normal conditions in the deep, not to say deep, water levels like 20 meters or 30 meters. But their maximum growth condition is 30 degrees Celsius. So these bacteria has colonies in 20 to 30 meters water columns, and they release the captured solar energies as a heat, and they warm up to each other to keep the natural environment at 30 degrees Celsius. Okay. If we change the carotenoids which are bound to this light harvesting complex so as that the number of the conjugated double bonds decreases, we can increase, we are successful to increase the efficiency of energy transfer from carotenoid to bacteriochlorophyll up to 65%, but not 100%. And what we need to have is systems that has 100% efficiencies and also all the colors. Okay. So especially for the green to yellow colors, relatively high efficiencies, but the orange to red is not very efficient. How we conquer these problems? Then what I have found is, okay, that bacteria is living in a very cold place. Why don't we go to the warmer place? And then I found Okinawa Mozuku. Okay, this sea in the summertime in Okinawa area, sometimes the temperature of the sea water goes up to 30 degrees Celsius. Okay, and in this Okinawa mozuku, they have a very special light harvesting pigment protein complexes called the fucoxanthin chlorophyll AC proteins. And in this case, they have this type of very complicated structured carotenoid, which has the RN group and also the carbonyl groups. In this case, since Okinawa mozuku, have you ever eaten Okinawa mozuku? It's the brown colors, right? That is the brown algae. Why brown is carotenoid is red and chlorophyll is green, so brown, green and red, brown. So in this complex, they have a red carotenoid, but the efficiency of energy transfer from carotenoid to chlorophyll is 100%. So this is the key. If we get into the deeper insight of the electronic structures of these unusual carotenoids, what we have found is they have ICT, intramolecular charge transfer type excited states, that is conjugated to the dark S1 state. And this state can make it possible to exceed the efficiency of energy transfer. Okay, so this is the key. Okay, so far, I have talked something about each pigment and each individual complexes, but actually in the natural light harvesting systems in purple bacteria, they are composed of the many pigment, many light harvesting complexes, and how they organize is like this way. This is electron microscope observations of native photosynthetic membranes of Bros viridis that has only LH1 reaction centers. And as you can see, there is a beautiful arrays, like a two-dimensional crystal types, LH systems. So they are so nicely, densely packed. But actually, in the natural systems, there is other types of photosynthetic membranes, which has both LH1 and LH2. And now we have good technologies to look at how they are arranged in the real photosynthetic membranes using high resolution AFM. And now we can see like this way, we can distinguish each complexes like this way. And also, we can make artificial type photosynthetic membranes, which we intentionally incorporate different type of the pigment protein complexes into the lipid barrier systems. Okay. How we can do that is like this way. So we isolate the LH2 or LH1 reaction center complexes from the different type of the photosynthetic bacteria, or we can modify the structures of these light harvesting systems, as I showed you before, like deassociation or reconstituted methods. And then we introduce both of them into the artificial lipid barrier systems, and then decide the precise arrangements using AFM. So this is how this view, this movie shows how these light harvesting pigments are arranged in not the natural. This is artificial photosynthetic membranes, and then we can now make much more deeper insight and make a much more deep research to reveal how the arrangement affects the overall energy transfer. Okay. This is very important because if we want to make a device, okay, which can be conjugated with some other catalysts. Okay. Okay, so that is the story about antenna systems. And how about the reaction centers? This is, the precise structures of the reaction centers has been decided sort of time ago. In 1985, the first crystal structure has been reported, and year 1988, they were awarded a Nobel Prize in Chemistry. Michael Dyson, Prof, and Huber, 3 German professors. And yeah, they have a very nice, this is an overall view, and this is the extraction of the pigment parts. They have a nice, two-fold type arrangements. There is a special pair bacteriochlorophylls, and monomeric bacteriochlorophyls, and pheophytins and quinones. And once the energy is captured by the special pair, within the 200 picosecond, the electron is transferred to this site. And interestingly, this two-fold type arrangement is conserved all the natural photosynthetic systems, not depending on the classes. Okay, bacteria, algae, higher plants, same, two-fold type structures. So what we can learn from here is electron transport reaction within the protein is not so easy. So probably this is one of the possible good solutions for the arrangements of the pigments for the electron transport. So in other words, we already know the good designs to produce electromotive force. Okay, in the last part, I will briefly explain what is going on these catalytic sites. Important thing is electrochemistry. Okay, to split water, we need 0.82 volt for the potentials, but actually we need to add 0.4 volt to overcome, the reactions. So 1.22 or 1.23 volt are necessary to split water electrochemically. To compare this amount to produce hydrogens from protons and also produce methane, methanol, carbon monoxide, and is not very large. Why is that? This is a very interesting chemistry point, because this is called as the mechanisms of the proton concerted multiple electron transfer. Okay, if you directly produce something from CO2, you need to use much more potentials. But if we concertedly can react the CO2, the potential is not as high as the water splittings. So in other words, the reduced catalytic sites reaction is relatively easier than the water splitting on the point of view of the electrochemistry. But actually, this is clearly mentioned in the very nice review paper by Professor Etsuko Fujita. She says an alternative and more favorable pathway is to reduce CO2 through proton-assisted multiple electron transport. That is the key. Okay, so we need to find good catalysts which has this kind of mechanisms. And actually, my good friend, Professor Osamu Ishitani at the Tokyo Institute of Technologies, he is already been very successful to reduce CO2 to produce CO using this type of rhenium coordination compounds. At the 2008, he has achieved the 59% of the efficiencies, but this year, this yield exceeds 85%. So for CO2 reductions, we now have the state-of-the-art technologies. But unfortunately, rhenium itself is more precious than gold. So this is another issue. How about the water splittings? Next week, Nobel Prize in Chemistry. This guy may have, Professor Fujishima. And Japan is always leading the research field of the artificial photosynthesis, I believe. And the original work which has been published by Honda Sensei and Fujishima Sensei is very famous Honda Fujishima effect using titanium dioxide type catalysts. And we have a recent remarkable progress, especially in water splitting. That is, the one is the structural determination of manganese calcium clusters. That's the oxygen evolution centers in PS2. And this structure may give us a good hint to develop and search for novel catalysts. And combination of metal oxide and metal oxynitride semiconductor system and co-catalysts another good candidates for the water splittings. And first catalytic cycle by way of ruthenium oxide also reported. And searching the catalytic activity using cobalt oxide as well as cobalt vein. And this is a good paper published by the group of Professor Kazunari Domen. They can combine two types of metal oxide and metal oxynitride semiconductors with co-catalysts and making this type of the Z-scheme like natural photosynthesis. And they are now successful to split water with apparent quantum yield of 6.3%. This is one of the state art. The other one that has been reported by the group of the Lichensund in Sweden. This is a very well-known, types ruthenium coordination compounds, but molecular ruthenium catalyst with water oxidation activity comparable to that of the photosystem 2 has already been achieved. And this is the manganese calcium cluster structures, which has been determined by the group of Professor Nobuo Kamiya and Professor Shen. And we referred to these structures, and now we are synthesizing this type of manganese coordination compounds. And right now we are in the stage to make to determine the activity or the mechanisms of the water splittings. Okay, this is the summary. Nature had evolved fascinating light harvesting and charge separation processes in photosynthesis. And I think the hybridization of natural photosynthetic antennas and metal oxide catalysis can be a possible approach to achieve artificial photosynthesis for the production of solar fuels. And there are some nice state-of-the-art technologies that facilitate water splitting and CO2 conversions. Most important thing is proton-concerted multiple electron transfers is essentially important to produce solar fuels. But the issue is how shall we do that without knowing detailed mechanisms in physics sense? So the collaborations with chemists, physicists, biochemists are very necessary. Before closing my talks, I would like to thank all these founding agencies for giving me a huge amount of money to perform the experiments for the last 10 years. And this is some part, now in the best year, I have got 50 academic staffs with me. Okay, 50 academic staffs, 100 papers per year. Thank you very much for your attention.